In chapter 12, we are now going to look at antimicrobial treatments. We talked about ways to control microbes in chapter 11. Now we're going to learn about how to treat them. So first of all, the main principle of any therapy is to administer a drug to the infected person that destroys the agent, and then the key is without harming the host. Unfortunately, the perfect drug does not exist. So some characteristics of the ideal drug, again, harmful to the microbe, not harmful to the host. It is microbial-cidal. Again, that means to kill rather than just inhibiting the growth. Soluble in fluids, because all of our cells and tissues have lots of fluids around them, so we want them to be able to penetrate. It's potent for long enough to act, but it's not broken down. And not broken down or excreted prematurely. So we don't want to have to keep administering the drug every hour or every few minutes. So it needs to last long enough within the body to have an effect. And so also that it has a slower, non-existent development of microbial resistance. So again, the idea is that microbes wouldn't be able to become resistant to it. It complements or assists the activities of the defense, so our immune system and things it's already doing. So how could it help it to work? It remains active in the tissue, so our cells aren't breaking it down. It can be easily delivered to where the infection is, and it doesn't disrupt the host health by causing allergies or predisposing to other infections. So some of our terminology, prophylaxis. We're hearing more and more about this term. So this is a drug that prevents an infection. So somebody's not already infected, it's meant to prevent it. A good example of this, people who have a knee replacement, they have prophylaxis antibiotic treatment before they go to like the dentist or to other places so that it prevents a potential infection. Then antimicrobial chemotherapy. Now, typically whenever we think about chemotherapy, we automatically go to cancer, but chemotherapy is any chemical treatment. So any use of a drug to control an infection. So again, try to keep in mind chemotherapy is not, it's just any drug, not just for cancer. Antimicrobials, these kind of the all-inclusive term here for anything, any type of drug, that, regardless of what type of microbe it's targeting. And then antibiotic, a little bit more specific. It's a substance produced by natural metabolic processes by some organism. So for example, the first antibiotic was penicillin, accidentally discovered because it was a chemical made from a fungus. So an antibiotic is something made from some other microorganism that's used to treat other microorganisms. Some more terms, semi-synthetic drugs. So these have been chemically modified. They were originally isolated from a natural source, so again, like an antibiotic, but then it's been slightly modified in the lab, so it's like half synthetic, half natural. Whereas synthetic would be entirely chemically produced in a laboratory setting. And then types of drugs, we have narrow spectrum and broad spectrum. So as the name kind of implies, narrow spectrum are antimicrobials that have a limited array. So they're only effective, say, against one type of species or just on gram-positive bacteria. Whereas broad spectrum, these are effective against a wide variety. So maybe both gram-positive and gram-negative bacteria. Now, starting the treatment, there are three main things that need to be known before we start. So first of all, identifying the organism causing the infection. So that's where knowing, again, is it gram-positive? Does it have something, does it have flagella? So identifying what the microorganism is. And then determining the degree that microorganism is susceptible to the drugs, and then the overall medical condition of the patient. And that can include things like pre-existing medical conditions, any history of allergies, especially like with penicillin. Do they have liver or kidney disease that will alter the metabolism or excretion of the drug? And then whether they're infants or elderly, as well as pregnant, those are all things that could change or specify which drugs can and cannot be used. So the art of kind of choosing this, so even though we have all of that information, the final choice isn't always straightforward. So we have to think about synergistic and antagonistic effects of drugs. If multiple drugs are being used, or if the patient's on other drugs, are they working together or are they offsetting each other? And also a big one is how they affect that human microbiome. Again, how does it affect all the good bacteria, all the other microbes that naturally inhabit the patient? So some of the drug microbe interactions, so selective toxicity, this is a core concept here. So we want the antimicrobial drugs to kill or inhibit the microbial cells without damaging the host. So selectively toxic to the microbe, not to the host. So the best drugs block the actions of synthesis of the microorganisms, but not the vertebrate cells. So for example, penicillins block the synthesis of the cell wall. So that's only in bacteria. So that doesn't interact with our cells because we don't have a cell wall. So finding targets unique to the microbe is important for selective toxicity. There are a few main ways in which these drugs work. So cell wall synthesis is one of the main ones, again, that's a unique target to them. Inhibiting DNA or RNA structure, that one gets a little trickier because we have DNA and RNA. Inhibiting protein synthesis, this one's a little easier because remember those ribosomes of prokaryotes are smaller than those of eukaryotes. But if we're talking about a fungal infection where we're talking about a eukaryotic, that gets a little trickier. Interfering with the membrane structure, again, gets a little trickier because we have cell membranes as well. And then inhibition of folic acid. That is something only bacteria make, so that's a nice target because we don't make folic acid. We take it in our diet. So just kind of a more visual here of those. Again, targeting protein synthesis by ribosomes. This is a very common one because, again, they have a slightly smaller structure than what our ribosomes do. So by inhibiting the ribosomes, it inhibits the enzymes that work so they can't make their products needed to survive or to go through metabolism. Again, folic acid synthesis is something unique to some bacteria, so blocking it prevents them from functioning properly. So like sulfa drugs, that's how they work, is by inhibiting that. Cell wall inhibitors, we've already mentioned penicillin. This is mainly how it works, is by either inhibiting the synthesis or breaking it down. The cytoplasmic membrane, again, loss of permeability, just kind of poking holes into it. But again, this gets a little trickier because we have cell membranes. So as you'll see, not as many options here under this class. And then DNA and RNA, so inhibiting replication. So they try to inhibit enzymes that are somewhat unique to bacteria, where they have a little different form than what our enzymes for replication are needed. But again, you don't see a lot of options because there's not a lot of selective toxicity. So quinolones and rifampin are ways of inhibiting DNA or RNA. But again, as you'll see, ribosome inhibition has the most we have because that's a nice target due to that difference in size. So the effect of these, at least 5% of taking them exhibit some type of severe adverse reaction, and that can be direct damage to the tissue. So again, unfortunately for them, it's not just toxic to the microbe, it's also toxic to them. Allergic reactions, especially again thinking about penicillin. And then the other one is disrupting that normal microbiome or that normal flora. And that leads to what's called a superinfection. So this is where by inhibiting the normal microbiome, it lets other pathogenic bacteria take over that are normally inhibited by our normal flora, by that microbiome. So that's called a superinfection, where you get another infection due to inhibition of that normal flora. Yeast infections are really common after antibiotic treatment because of that. It disrupts the vaginal normal flora and allows the yeast that are normally there but kind of kept in check, lets them grow much faster. And then a little bit more severe antibiotic-associated colitis is due to changes in the gut normal flora that allow Clostridioids difficile to start growing more than it normally would. So just some of the spectrum activity. So again, thinking about narrow spectrum, so like tropomycin and polymyxin, they have a pretty narrow spectrum, only kind of effective against Mycobacterium that cause tuberculosis, whereas our broadest spectrum here would be tetracyclines, as you can see, used to treat lots of different, so gram-negative, gram-positive, chlamydias, rickettsia that causes Rocky Mountain spotted fever. So again, this is just kind of showing how different these antibiotics can be as far as their spectrum. Now, if we start looking at some more specific actions here, so drugs that target the cell wall, again, these are mainly going to be our penicillins, and they have what is called a beta-lactam because of this group called a beta-lactam group. It's a chemical kind of group. I'm not concerned that you know what it looks like, but just understand all these different types of drugs share that beta-lactam structure, so they're grouped into the same category. And again, they all alter the cell walls. So for example, penicillin inhibits the synthesis. So it prevents the cell wall from being formed, all that peptidoglycan coming together, so they can't multiply. So it doesn't necessarily kill the ones already there, but they can't multiply to cause an infection. Now the other ones that alter the cell wall, so cephalosporins, they also have that beta-lactam ring. And act similar to penicillin. They inhibit the synthesis. Carbapenem, this is reserved just for use in hospitals when other drugs aren't working. So if we have a resistance or an allergy to some of the other ones, it's kind of more of a last resort. And then just a few others that alter the cell wall. So bacitracin, isoniazid, and vancomycin. Now some, again, our biggest category here that alter protein synthesis. Again, all of these are broad spectrum because all bacteria have ribosomes, so they're able to target kind of any bacterial infection because they all have these ribosomes. So just some of the categories. So aminoglycoside like streptomycin, it's inserted in the small ribosomal subunit, and it causes abnormal proteins to be formed, so they don't function right, so the cell can't use them. Tetracyclines also alter the ribosomes, but they block protein synthesis or translation. It prevents amino acids from coming in. Microlids like erythromycin, they inhibit the ribosome from moving during the process of translation, so it blocks that translation process. Some other ones that inhibit protein synthesis, streptogramin, that inhibits the larger subunit, again, inhibits that protein synthesis or translation. Isonazolidinones, these are synthetic, and they inhibit initiation or the beginning of protein synthesis. And then the pleuromutilins, they inhibit the translocation, so the movement of the ribosome down the messenger RNA to make that protein. Then those that alter the folic acid, again, as I mentioned, these are the sulfa drugs or sulfonamides. So what they do is they block the enzyme that's needed for synthesis. So here's our enzyme that's needed to make folic acid. The sulfa drugs bind where the substrate should be binding, and it blocks it. So then that substrate can't come in, so then the enzyme isn't helping to produce the product. So they can't make folic acid, and folic acid is needed to make their DNA and RNA, as well as some of their amino acids. So by blocking that enzyme, it prevents that synthesis. Then as far as targeting the DNA or RNA, the quinolones, this one, so fluoroquinolone, for example, like ciprofloxacin, it's very potent, again, broad spectrum because most bacteria replicate the same way. It inhibits an enzyme called helicase that unwinds DNA. So in order for DNA to be copied, so the cell can divide and multiply, helicase comes along and it essentially breaks these bonds between the DNA and it separates them into two strands. Well, what this group of drugs is it blocks essentially the scissors, so the DNA can't unwind, and therefore it can't go through DNA transcription and it can't be replicated. Then some that alter the membrane, so the polymyxins and damptomycin. Again, this is narrow spectrum peptide, just meaning they're protein antibiotics. They have a really kind of unique fatty acid component to them that kind of acts like a detergent. So just like the surfactants in our controlling microbes, like our detergents, this drug actually kind of acts like that. So it binds and inserts itself into the phospholipids, distorts their shape, and causes them to leak fluids and makes them really impermeable, or really permeable. Now, one of the issues that would come back to those biofilms, that community of microbes, again, the first microbe secretes kind of that sugary slime layer or capsule, and then lots of other microbes start attaching to it. So just to put it in perspective, it's 1,000 times less sensitive to antibiotics whenever they are in a biofilm than if they're free living. So just to kind of put that in perspective of how much harder it is to treat organisms in a biofilm, because there are different phenotypes and different characteristics whenever bacteria are in a biofilm versus by themselves. So whenever we're thinking about treating them, they need to interrupt what's called quorum sensing, which is how they kind of recruit other players and other microbes to their community. Damptomycin is really good at this as far as treating that. Again, it inhibits the replication and can be used, especially pre-treatments are important to kind of break down the biofilm and then add the antibiotic. Now kind of switching to fungal infections. Now these get a little bit more tricky too. We talked about how hard it is to treat viruses because they don't have very many targets. Well, the problem with fungi is that they are also eukaryotic, and we are eukaryotic. So those create their own little problems. So first of all, antibacterial drugs are not effective against fungi, just like they're not effective against a virus. So the similarities though between fungal and human cells mean, again, we have less targets that are unique to fungi. So the drugs can be toxic to fungi, are also capable of harming human tissue as well. All right, so if we look at some of these categories, so microlids, these alter fungal membranes. Again, their membranes are similar to ours, though, so this becomes, somewhat finicky as far as selectively targeting fungal membranes without ours. But just like the ones we talked about with bacteria, whenever they kind of poke holes in them, then the membrane's it becomes more permeable. It's more likely to rupture into lice. Now the azoles, they interfere with sterol synthesis. Now this is a unique compound that is found in their cell membranes. It's a little different than what ours is. So that's a nice target. The echinocandidates, this is probably the most selective because it targets fungal cell walls. So fungi do have a cell wall that we don't, so that's a nice target, just like bacteria cell walls. Now they're also not the same though. Bacteria cell walls are made up of peptidoglycan. Fungal cell walls are made up of a carbohydrate called chitin. So penicillins don't treat fungi because the cell walls are made up of different things, and vice versa. And then finally, the amines. These inhibit enzymes that are critical for that ergosterol or that sterol synthesis. So kind of similar to the azoles, but it's just inhibiting its synthesis rather than directly interfering with it. Then moving to our protists and to our helminths. So the anti-protozoan anti-helminths, again, these are also eukaryotic, so it creates some problems. The main one that we talk about here is anti-malarial drugs. Now, thankfully in the US, this malaria is not a huge problem, but for those that are traveling, it can become an issue. So quinine, is the main treatment for it. However, it is starting to be replaced by chloroquine and promequinine, which are a little less toxic to humans. However, there's not a single drug that can, universally affect every species of the Plasmodium that causes malaria. Typically, artemisinin combination therapy is used because it treats, it's a combination therapy that treats different types of malaria. So the treatments for other protozoan infections, kind of the go-to generic drug is called metronidazole. It inhibits a lot of protists that are there despite kind of the species. So it's kind of a go-to as far as protists, except for those for malaria. The anti-helminths, again, these are parasitic worms like tapeworms. There are a few. Ivermectin, especially if you are familiar with, like, have animals deworming. This is the main one that is given as a dewormer, is ivermectin. It can also be used in humans, but then a couple others, albendazole, Pyrantel, and Parasequintel are also ones. But again, ivermectin is usually the most common one we see. And then again, our antivirals, as we already mentioned, again, really hard to target because they're just protein coat and nucleic acids. Sometimes they have some enzymes in there. So the three modes of action is just preventing penetration. So altering their spikes, preventing that receptor binding or uncoating that has to happen. So if we can prevent it from getting into the cell, we can prevent the infection. Other ways is to prevent replication or transcription, especially like with HIV, where it's a retrovirus. It has a specific enzyme it carries with it. So that's a nice target. Some other viruses have unique enzymes, but again, most of them use the host cell. So there's not a way to target the virus. And then finally, just preventing the maturation. So putting the actual DNA inside of the protein coat, that capsid, and preventing the spike. So if we can prevent kind of the assembly of the virus, then that can prevent the infection as well.